I am Somak Datta. I'm a faculty member in the Department of Statistics, and I'm substituting for Caroline today. Today we have Dr. Roland Schafleitner and Dr. Stephanie Greene. These seminars are possible because of generous support from these two institutes, so the HIPS, and which is HIPS is funded by the USDA NIFA, and the AIRA, AI Institute for Resilient Agriculture, which is funded by both USDA and NSF. Our first speaker today is Dr. Roland Schafleitner. He's presenting from Taiwan, I think. So thank you, Dr. Thank you, Dr. Roland, for, it's probably midnight there. So a special thank you for joining us today here. Dr. Roland holds a PhD in biochemical technology from the University of Vienna in Austria. After 6 years at the International Phenomics Center in Peru, he joined the World Vegetable Center in 2010 as head of molecular genetics, with the task to produce genomic tools for crop biodiversity research and vegetable breeding. While genomic tools for plant breeding became more and more accessible, plant phenotyping remained a bottleneck for the analysis of genetic basis of plant properties. For this reason, Dr. Roland got interested in methods and tools that make phenotyping affordable. Today, he will talk about how Phenomics measures trait variation in large vegetable gene bank collections and helps breeders to identify new donor materials for interesting traits for breeding. So, Dr. Roland, if you could share, let me stop share my screen. If you could share your screen and the presentation.
Thank you very much for the introduction. Before I go to the topic, please give me a second to introduce you a little bit to the World Vegetable Center. I don't know whether you have heard from us before. As the name says, we work on vegetables, and we work on vegetables in order to work for healthier lives and more resilient livelihoods through greater diversity in what we grow and what we eat. That means we are conducting research, we are building networks, we are performing training and promotion activities, and all that to strengthen the role of vegetables for improved health and global poverty alleviation. Global poverty alleviation because we are working for smallholder farmers, and we think that vegetables, or we know that vegetables are a better method to earn money than on a small farm than, for example, with cereals. Generally, with vegetables, smallholder farmers earn more. That's why we use our vegetables for both as a vehicle to improve nutrition and to earn money. My task at the World Vegetable Center is to oversee the research around vegetable diversity and improvement. In this division, we hold probably the world's largest public sector gene bank, with now some 65,000 vegetable accessions of 330 different vegetable species. And from these accessions, we try to pick out traits that we can use in breeding, but also accessions and species that we can develop into new varieties directly from the gene bank in order to diversify agriculture. This collection, of course, is also available for public and private sector users at a very, very low fee. We have breeding programs, active breeding programs for tomato, pepper, for 2 cucurbits, some legume crops, and for a crop group that we call traditional African vegetables. This is a crop group that comprises a few species that are particularly nutrient dense. We always talk about the gene bank, but in fact, we have 2 gene banks. We have the gene bank at the World Vegetable Center headquarters in Taiwan. This is the one I introduced you before. The 450 species here on this slide is wrong. It's only 330 species. And we have also a relatively small but very fine gene bank in our center in Eastern Southern Africa in Tanzania, where we hold 2,700 accessions of African traditional vegetables. This is the biggest public sector accession of this public sector collection of this kind. These are a part of the crops that we are conserving and promoting for use in our gene bank. The biggest collection we have is the one for soybean, and then follows tomato, capsicum, some Vigna species like mung bean and suki bean, then eggplant, some cucurbitaceae, okra, onion, and traditional vegetables. When we talk about gene banks, well, there are different qualities of gene banks. Gene banks very often start as early as seed depositories, where seed is stored and conserved as well as possible. Once information is added to the seed, like inventories and passport data, the seed becomes already more useful for users. Then we can talk of a real gene bank. When we add more information, like standard descriptor or evaluation data, it's even getting more valuable for users. If we add then even genotype data or trait information, then I wouldn't call this a gene bank anymore, because we are not just distributing seed. In this case, we're also distributing information with the seed. And like that, the seed becomes extremely useful for breeding and for research. And my credo is that if we want to conserve genetic resources, we also must promote their use. I think the best conservation method for genetic resources is, in fact, their use. One way to make genetic resources more useful, in my view, is to make core collections. For some of our germplasm collections, we have many accessions, 10,000, 20,000 accessions. This is very often by far too large for scientists to screen these large collections for useful traits. So more and more we produce core collections. These are smaller collections that represent the diversity of the large collections in about 300 or 400 accessions. For this purpose, we start with germplasm inventories. Then we do genotyping in order to measure the genetic distance between the accessions. And based on the PCA and the very simple analysis, we select accessions that represent best the biodiversity or the genetic diversity of whole collections. We have done this in collaboration with a EU-funded Horizon 2020 project, G2P for tomato, pepper, and eggplant. And in the scope of other projects, we have done a similar job for our large mung bean collection and for our amaranth collection. And these small collections are very practical. We can relatively easily distribute them to users, and we can evaluate them across different environments. And like that, we can find the traits that the breeders are actually seeking for their programs. But still, phenotyping is by far more laborious than, for example, genotyping a collection. In order to overcome this bottleneck, our first investment in phenotyping was this field phenotyping system, here shown with our pepper core collection. This is nothing else than a gantry that moves over the field. On the gantry, there are mounted two sensor heads, and these sensor heads are scanning the plants. They are emitting near-infrared light. The plants, or everything that is between the sensors and the soil, will reflect this near-infrared light. And from this reflection, the sensors are building up a 3D data cloud of the plants. In my opinion, these 3D data clouds are much more easy to analyze than pictures. With a suitable software, we can automatically, without any human intervention, extract data like plant height, leaf area, digital biomass, and much more. And in parallel to the near-infrared light, these sensors also have RGB flashlights. So over each data point that represents this data cloud for the plant, we can overlay a color value that allows us to calculate some physiological parameters for the plants like NDVI and others. So this is a very simple phenotyping method that works at very low cost, but provides us with a wealth of data. And what I like particularly of this data are that these data are collected over time in a non-destructive way. So we get growth curves, or we get a depiction of the development of the plant, and instead of just point values at a few time points. So this is a real great tool to literally watch the plants growing. And one of the tasks of this phenotype, of this field phenotyping station, was to characterize the response of our pepper core collection to heat stress. We did this analysis over 3 seasons, 2 hot seasons and 1 cool season. And in parallel to the field assessments, we were checking some traits by hand, like yield components, and we also have applied flow cytometry to automatically analyze activity of pollen, which tends to become less active under heat stress. Taking together, we could associate many traits either with each other, or we could find several traits that contribute to yield. There were a few traits where it was absolutely logical that they are going to contribute to yield, but the interesting thing, in fact, were some surprises. And one surprise to me was that, in fact, in pepper, leaf angle and leaf angle change is an important factor for tolerance to heat. Here, the bars in blue show the leaf angle change under cool temperatures. It shows, in general, that the leaf angle are large. That means the leaves are expanded during cool temperatures, and the plant can do very good photosynthesis. Under heat, the leaves move and the angles become smaller. And it depends on the genotype how small these angles get. And interestingly, this trait is quite well associated for yield. So if we want to breed for high-yielding pepper under heat stress, taking into account leaf angle and leaf angle change is quite useful. And this came for us as a surprise. We know from other plants that the leaf angle is playing a role. We did not know this about pepper. We would have never seen this with manual measurements. It's only by using this field scan device that we were able to see this feature in pepper. The measurements that we get from this Phenospecs field scan, then we are trying to analyze and to make more sense out of it. We have developed some machine learning tools, or let's say better, we have adopted some machine learning tools in order to understand the traits better and in order to see better the variation of the traits in the germplasm. For example, we were asking ourselves, at which temperature does this core collection in average react in terms of some parameters like senescence, vegetation index, or chlorophyll index? And we found that there are some typical temperature thresholds where these kind of parameters show the biggest changes. This helps us to determine at which temperature these kind of traits might play a role, and at which temperature we have to look in order to see a difference between the more susceptible and the more tolerant accessions. And of course, this kind of analysis also shows us much better which of the accessions reacts in a more sensitive way and in a more tolerant way. So this is a very practical, or it became a very practical tool for us to analyze relative large germplasm collections and to find trait variation and tolerant germplasm at the same time. During the last 3 years that we have this equipment, we have run some 4 larger trials. We checked our Amaranthus tricolor collection for some growth traits over 2 seasons. The results are just being finished to be evaluated. One paper is submitted, but what we see here is mostly useful for horticultural purposes, less for scientific purposes. Then the capsicum core collection that I have already described very briefly, which gave us very interesting insight for breeding, but also some new information about traits. Then we have tested flooding tolerance in an okra core collection over 2 seasons. And this allowed us to isolate, to identify some okra accessions that continue growing and yielding under flooding stress, and others that recover very rapidly after flooding. Flooding stress is quite a frequent stress in tropical environments, where during the hot and rainy season, flooding is quite a frequent event. And okra is one of the crops that can grow during this season. So a flooding tolerant okra is quite in demand by farmers. And just now, we start to analyze our Vigna radiata mungbean core collection for plant architecture, harvest index, and other traits. So as a resume, I would like to conclude from these experiments that this kind of phenotyping is ideal to generate information to gene bank accessions. And I think that this information really is adding value to the collection. We don't hold and distribute only seed from now on. We have information about the performance of this seed, of the plants that grow out of this seed. So it is really an added value. And then, of course, as I mentioned already, this kind of experiment allows us to measure trait variation in the collection and to identify sources for tolerance traits, for example, of other breeder desired traits for breeding. It leads to a better understanding of traits and how different traits are working together. And it is the basis, it brings the phenotypic data for genome-wide association mapping and for crop models. We have already started to work also on this field and have developed crop models for flowering in mungbean, for example. And we have performed already the GWAS around the heat stress tolerance traits on pepper. And this is a work that is going on and on now at the World Vegetable Center. And I think that at this time, my 20 minutes are over. I would like to thank you for your attention. And I would like to thank those colleagues who have contributed to this work. There is, first of all, Derek Barchinger, our pepper breeder. He was responsible for the 3-season pepper trial, and this was the most successful phenotyping experiment that we have had in the last 3 years. Then Ms. Lin Yaping, our bioinformatician, who is, through her hand, holds practically all the data that we are generating in our group, and she develops the analysis methods. Now her hobby is machine learning and artificial intelligence. Then Ken Hoshikawa, he is our seconded scientist from Cherkas, Japan. He is a very good geneticist and is also very firm in plant physiology and is contributing strongly to these experiments. And Ms. Lin Chen-Yu, who is managing the laboratory and the field experiments in our unit. I also would like to mention Kevin Fernandez and his team from Phenospecs, who are always helping when there is a problem with the phenotyping, when we need new ideas how to analyze the data, or when a sensor breaks, they are reacting extremely rapidly to any problem. That's why we could run these experiments over 3 years practically without interruption. And I would like to acknowledge the funding agencies. And before I conclude, really, I would like to invite you to the World Vegetable Center. We are considering ourselves as an open science center. We are open for visiting scientists. If somebody is interested in vegetable research, in phenotyping, in genotyping, in pathology of vegetables, in vegetable gene bank, please visit our webpage and consider to come to us and to work with us and to pass a sabbatical year with us. Or if you are a student, why not making a PhD thesis with us? Please check our webpage. Thank you very much for your attention.
Dr. Roland, for the great talk. There are time for questions. Anybody in the audience here have any question? Oh yeah, go ahead.
Hi, yeah, thank you for the talk. It was really cool. I work on maize, so it's cool to hear about other plants out there. But one question I had was, you mentioned these like core collections of diversity. Can you only construct those if your vegetable has an annotated genome? And about how many different vegetable species do you have that do have an annotated genome?
Well, most vegetable species in the meanwhile have an annotated genome, but the core collection does not depend on that. We can also genotype, in fact, without an annotated genome, and we have started more and more to work with markers without reference sequence. We have several reasons for doing this, because our collections are very diverse. If we would use a reference sequence, we would lose much important information. And very often you don't have the reference sequence from the very interesting, super tolerant and super resistant lion that you find in your gene bank. And when you work with a reference sequence, you are generally blind for those genes that make these plants wood. So we prefer to get confusion through paralogs and perhaps are not able to use our markers in the GWAS, but we can use these markers in order to look a little bit deeper into the diversity. What we're also doing with Mangwin now at the moment is we won a grant from Illumina. I think it was 20 terabase pairs of sequence information, and we use the sequence information to sequence a large part of our collection. And that makes us a bunch genome, at least a bunch genome with Illumina sequences, which is quite good. It will take still some time to finish that, but just to your question, reference sequence, I think, is a thing of the 1990s. Now we work without. Thank you.
See you. And okay, are there any questions from the audience, online audience?
Hi, Roland. This is Sean McEachran. I work for Sensei Ag. Sorry, Andrew. Very interesting. I wasn't aware of your group and the collection. It seems very impressive, and I really appreciate the strong emphasis on phenotyping. What sort of process do you have around collecting, or if we were interested in seed for various traits like drought tolerance, etc.? What's the process there, and how do you distribute it globally if you're based sort of in Taiwan? Is that a difficult process?
Our germplasm collection is the biggest part in the public domain. That means that we are distributing this material under a standard material transfer agreement, like it is used or suggested by FAO. So it is very easy for you to get this material. This material has been either collected before the CBD came into power, or we have the right to distribute the material because it has been collected with agreement of the National Nagoya Protocol responsible point, or I don't know how you mentioned that. So from this side, you have access to our collection. If you're interested in specifically drought-tolerant material, I suggest that this material is brought somewhere else than away from our beautiful island, because we are a very green island. It rains a lot. So, for example, for amaranth, we are using not even drought or rainout zones here on the island. We are shipping the material to South Africa, and ARC in South Africa is working in rainout zones, because even when I build a rainout zone here from the subsoil, I get water. That's just the example for drought tolerance. But as I mentioned, we are an open science center, and we welcome very much colleagues coming to us, working with us, and doing the screenings here. Or we help you to decide about which material you would need to make your screening in your institute. So both ways are possible. For some crops, I mentioned them: tomato, pepper, eggplant, amaranth, mung bean, okra. We have core collections that are very practical to ship around. And we know already about the diversity we have in these core collections and how much of the genetic diversity, at least, they represent of the whole collection.
Excellent. Yeah, very interesting. And I like the idea about maybe students as well, right? Maybe sending a student over to look at whatever
Getting
trait.
students.
Yeah.
It's a little bit difficult now still with COVID, but we are opening up.
Okay. All right. Thank you very much.
Thank you.
Okay. In the interest of time, we'll just take a question from Andrew.
Hi, yeah, thank you very much. I'm curious about the phenotypic information you generate and how you think that's being used by sort of your customers, for lack of a better term. Is it mostly to help them make more informed decisions about the material they're requesting? Or do you think any, say, public breeding programs are actually able to incorporate your phenotypic and your environmental data into their own models? How does the phenotyping translate to your stakeholder experience? Thank you.
Thank you very much. This is a very interesting question. We have now 3 years of experience in phenotyping. Before, we just did field evaluation, and you know the difference. This field evaluation was already translated into stakeholder information in form of our breeding programs and in form of distributing material that we knew that it has this or that property. Now, with this high throughput phenotyping tool, and we are now also expanding towards drone-based methods, and in about 2 years also controlled condition methods. And we are working with colleagues in Taiwan who also have invested in phenotyping, and we can interchange tools with them. So with the help of them, we can really isolate parts of core collections, and we can make these core collections fit for certain environments. Just for example, I did not show that. We have made an investigation of how a collection of 300 pepper accessions behaves over a gradient, a temperature gradient between 15 and 35 degrees. And as we had also genetic information or SNP information, we could produce something like a GWAS for certain heat stress tolerance traits across a whole gradient and temperature gradient across 300 accessions. You can imagine if somebody would like to have accessions that grow best at 28 degrees, with this kind of tool, we can practically identify them. And then we routinely run always an AMI over all these analyses. So we also identify materials that show broad adaptation. And we do this for 50, 60, 70 traits. The luxury that we have is that these 60, 70 traits are measured only by 2 stuff. We don't need more. The analysis then takes a little bit of time, but still is extremely quick. And we get this information. You were asking about the impact for our stakeholders. In 3 years, it's difficult to see impact, but just for the pepper core collection, we have distributed it last year, I think, to 30 recipients. And the mung bean core collection, we have distributed to about 20 recipients. And they receive also the information with that. And they can also narrow down then this 300 accession collection to perhaps only 100 accessions using this phenotypic data. But I really appreciate the question. It makes me think much more how I can make our collections better suited for our stakeholders. Thank you very much.
Thank you very much.
Let's thank the speaker once more, and let's move on to our next speaker. Dr. Stephanie Greene. So Dr. Stephanie Greene is a supervisory plant physiologist with the USDA Agricultural Research Service, located at the National Laboratory for Genetic Resource Preservation, NLGRP, at Fort Collins, Colorado. NLGRP provides secure backup storage of the USDA National Plant Germplasm System, this system collection, and other seed collections from around the world, making it one of the largest seed repositories in the world. Dr. Greene's research focuses on the conservation of crop wild relative species native to the United States. She received her bachelor's and master's from the University of Idaho and her PhD from Kansas State University. Dr. Greene has worked the last 30 years in the NPGS, and today she'll talk about the USDA Gene Bank conserving precious plant genetic resources.
Thank you for the introduction and for welcoming me into your lunch. I appreciate that. So I'm going to take a little bit of a different tact from Roland. He gave a really nice presentation on the phenotyping that's being done in his gene bank in the World Vegetable Center. And that is an essential part of gene banking. Again, I loved Roland when you talked about this idea of a biodigital repository. That is definitely what gene banks are moving to as quickly as we can. What I'm going to focus on today, though, is giving you an overview of the National Plant Germplasm System in general, and then focusing in on some of the challenges that we have, which are shared across the world by all gene banks. And I hope you'll come away with a better understanding of what gene banking is all about, what are our challenges, and what are some of the ways we're moving forward into the future. And I am gonna turn off my video because my internet sometimes is a bit flaky. So that just kind of helps things. Okay. All right, so the National Plant Germplasm System, which is our official term for the USDA gene banks, is housed in the USDA Agricultural Research Service. This is a part of the USDA. It's often called the research arm of the USDA. And the mission is to basically deliver cutting edge tools and solutions that support the well-being of people, particularly the food, through the food they eat. So we're focused on providing tools to support the nourishment of people, as well as providing tools that help sustain agroecosystems and natural resources. Of course, the bottom line is that we want to ensure the US competitiveness and the excellence of our agriculture. So plant germplasm is key to reaching these goals and to develop tools. Germplasm is essential to meet agricultural threats through this process of continued crop improvement. That's the bottom line. You need diversity to make improvements in crops. And plant breeders and other scientists need access to this material. And so gene banks are here to conserve and also make available this diverse germplasm, but also the information that's associated with it. So the National Plant Germplasm System dates back a long time. Basically, 1898, the USDA set up a department whose objective was to go out on collecting missions around the world to collect germplasm, which was then brought back into the United States and distributed around the country. And extension agents, government agencies, and farmers grew out this material and pretty quickly figured out what worked in the United States and what didn't in terms of the crops. It's essentially these efforts that occurred around in the late 1800s formed the foundation for the agriculture that we have today. Fast forward to 2022, and we now have the National Plant Germplasm System. Things have really changed in the germplasm world since the early days of plant introduction. We don't go around the world taking people's germplasm. Since the Convention on Biological Diversity, a country has the sovereign rights over their own materials. And it's not a simple matter of going into a country and collecting their material and bringing it back and growing it and producing it and commercializing in your own country. So the whole dynamics have changed. The National Plant Germplasm today, though, is one of the largest national gene bank systems. And we have very large collections of major staple crops across that are important to the United States and to the world. The material that we distribute is available around the world, and it's freely distributed. The collection is pretty large. We have about 600,000 accessions that represent over 16,000 species. Now, most of these species represent economic crops, but more and more we're getting a lot of wild material, either crop wild relatives or species that are important for revegetation work. The database that manages all this information for the entire NPGS is called the Germplasm Resources Information Network, or Greene Global. And this is the place you go to find out what kind of germplasm is available to you to support your research, and to get basically a better understanding of the collection and the data that's associated with our various accessions. So if we look back over the last 10 years, the size of the NPGS is stabilizing. Right now we have a few gaps that still need to be addressed, but for the most part, we're not growing that much anymore. However, the demand for our germplasm has been pretty steady. We will annually distribute about 250,000 accessions around the world. You can see, though, that we did take a dive in 2020 due to the pandemic, but our distribution is back up to how it has been in the past 5 years. So the NPGS is actually a network of gene banks spread across the country. We have about 18 gene banks that manage specific crop germplasm collections. For example, in Ames, you have the North Central Regional Plant Introduction Station. I'm sure that some of you have probably toured the Ames Gene Bank. It's one of our most impressive gene banks. They have about 54,000 accessions. That's where the maize collection is kept. They have the oilseed crops, amaranth, and associated pseudo cereals. They have a large vegetable collection and ornamentals. Out on the East Coast in Beltsville, Maryland, we have a laboratory who just focuses on Greene Global. So it's our database management unit that works on Greene. And we also have our plant exploration, our plant exchange office that manages our plant collecting program. And then there's my lab that's in Fort Collins, Colorado, and we're responsible for managing the safety duplicate collection for the NPGS. So we are a network of gene banks, USDA, we're a federal organization, but we also have a lot of partnerships with our land-grant universities. Most of our gene banks are located on university campuses. And we have had these partnerships for a really long time. It's an important part of our network. So what does the day in the life of a gene bank curator look like? Most of our curators have PhDs. They're responsible for a set of collections. And what they focus on is taking care of the collections. They're responsible for these collections. They're responsible for understanding them, for identifying gaps in the collections that need to be filled. Many curators will go out on collecting trips to fill gaps. They're also responsible for maintaining the collection. So making sure that the seed or clonal propagules are maintained correctly in storage so that the viability remains high. Every year, what they'll do is grow out a portion of their collection. We call this regeneration. What you can see in the background photo is the alfalfa collection being regenerated in Eastern Washington. The regeneration is done very carefully because we want to minimize any genetic change to our accessions. We want to maintain the genetic integrity. And so for the alfalfa collection, each accession is growing out under a cage that controls pollinators. And alfalfa is pollinated, it's cross-pollinated by bees. And so once the plants start flowering, and again, we have about 100 to 120 plants in each of these cages to minimize genetic drift. Once our plants start flowering, we'll actually put in leafcutter bees into each one of these controlled pollination tents so that we have good seed production. If you have, again, I think probably a lot of you have visited the Ames site, or I hope that you have. Ames has an amazing fields where they do a lot of controlled pollination work. And it's really fun to go out in the summertime to take a look at all of their efforts to regenerate material. Another super important activity that a curator focuses on is the documentation of the collection. Again, what Roland talked about is that it's not just the biological material itself, it's the information associated with it that makes it so valuable. And so curators will really focus on gathering data, they characterize the collection, they evaluate collections, they get this data uploaded into Greene where it's available to be looked at and used and queried on by our users. We're also interested in enhancing collections and of course doing research that support all of these priorities. The reason that I've got this colored the way it is that what we're looking at is sort of how we prioritize our activity as to what is the most important to the least important when your resources are limited. So the hot colors, red, are our most important activities. These have to be done to maintain our collections. As we have resources, we spread out and focus more on the activities that are cooler colors. So the National Laboratory for Genetic Resources Preservation, that's my lab in Colorado, is focused on storing for the long term security duplicate collection of the National Plant Germplasm System. And we also store other gene bank collections. Our vaults are large enough to be able to offer this service. And you know, you don't think about it, but it is incredibly important to duplicate your collections in another geographic location. Right now, the entire gene bank community is very concerned about Ukraine. We have not heard what is the situation with the Ukrainian gene bank. It's located in the northeastern part of the country. I visited in 2008. Their seed stores their seed is stored in glass bottles. We have been reaching out to find out if the gene bank needs any sort of assistance to move the collection, and we haven't heard anything. Fortunately, the Ukrainian gene bank has been duplicating some of their collections at the Svalbard Global Seed Vault that's in Norway, but the entire collection hasn't been duplicated. And Ukraine has some very important germplasm, particularly for wheat varieties. So again, it's important to back up your collections in a different geographic location. And the US is lucky that we do have our facility in Fort Collins to provide that role. Although even the USDA sends their material to Svalbard as well, to again get a third level of backup. Our lab is pretty big. Not only are we storing plant material, but we also house the National Animal Germplasm program, which stores and manages livestock germplasm, mainly in the form of semen. And we also are able to store important duplicate collections of various microbe collections that are important for agriculture. So just looking at the plant collection that we have in our vault, we have close to 1 million samples. Again, half that material represents the NPGS duplicate collection. We have about 82% of our seed collections backed up at Fort Collins, and about 20% of our clonal collections that are backed up in liquid nitrogen. We have some substantial collections that are just found at Fort Collins. And then in addition, we have 500,000 accessions where we're holding other gene bank collections, again, for safety duplication. And these include national collections. We have the Canadian collection, for example, but also international collections, for example, the Erie rice collection. And we even have about 3,000 accessions from the World Vegetable Center at our location. So in addition to storing all of this material, we also conduct research. We have 4 scientists in our laboratory. The focus of these scientists is on developing new preservation protocols. We also design and test strategies for exploiting genomic data to help us better manage NPGS germplasm. And we're also focusing on the preservation of crop-wild relatives, basically developing and validating strategies for collecting material, preserving, and using it. So there's some key challenges that are facing the NPGS. This slide illustrates how our budget has sort of plateaued over the last, since 2009, essentially. The budget has been adjusted for inflation. So what we can see here is that our actual real budget has been declining, despite the fact that our collection continues to grow and that our distributions continue to grow as well. So some of the challenges that we're facing include the need to expand our operational capacity. We need more storage space. We need more greenhouses. We need more equipment to better phenotype our collections. We also need to really address and get better at cryopreserving clonally propagated crops. So a clonally propagated crop is something, they're mainly fruit and nut crops. And the way they're held, because you can't, it doesn't make sense to store seed because again, they're clonally propagated and the seeds would, outcrossed out in the field, they're going to represent whatever's out in the orchard. And so what we are developing are ways of backing up this material by actually treating dormant buds or shoot tips with cryoprotectants, and then we can put them into liquid nitrogen where they'll stay for a long time. We've got material from the apple collection at our lab that's been there going on to about 35 years. And what we can do with these dormant buds and shoot tips is we pull them out of liquid nitrogen. We can actually revive them graft them onto an appropriate rootstock and get the buds to start growing again. The problem is that we need a lot of more resources, develop the technology to be able to do this. We're also entering into a time when genetically modified cultivars are entering into the national plant germplasm system. And this is brings a whole host of issues, mainly around not being, we need to be hyper vigilant about not letting transgenes inadvertently move into some of our germplasm. Again, our intent is to maintain the genetic integrity of each and every accession. And so we need to develop a set of procedures for managing these genetically engineered accessions and also controlling adventitious presence of GE traits. And then another challenge is also to go out and make those collections of crop wild relatives. The work I've been focused on is on crop wild relatives here in the United States. We have quite a lot of crop wild relatives in our own backyard. We did a gap analysis of 600 high priority crop wild relatives, high priority in that they're important to breeders. And we found that there's still a lot of work to be done. There are species out there that are critically endangered. 50% may be endangered, and that we have major gaps in our collections. And so we need to get out, make these collections on the ground, get them into gene banks so that they're available to be used by breeders, but also secure from things such as wildfires, all of the issues that climate change is bringing in. They're vulnerable species. Another challenge that we've been facing in the NPGS is just retirements. There was a wave of hiring, 30 years ago, and now everybody's retiring. We have a lot of work that we need to do to hire and train people to curate these collections, to take care of these collections, to ensure that they're managed in the best way possible so that they can continue to be used and are valuable now and into the future. So it's not all doom and gloom. It was really exciting. In 2018, the Farm Bill actually required the NPGS to develop a comprehensive plan for what is it going to take to carry out our activities the way they need to be carried out for the next decade. Now, this is really super exciting when Congress wants you to write a plan. So we all got to work and really itemized what it is we need to do the job right. So the plan has been completed. It's undergoing review, and sometime in 2022, it will be presented to Congress. And what we're hoping is these efforts with Congress looking at what it is the NPGS needs, that we will be able to, action will be taken that will enable us to strengthen the national plant germplasm system. We're hoping to see increased annual funding, expansion of our NPGS staffing. We want to expand our cold storage, our greenhouse space, and of course, expand our laboratory space. This would also include major efforts, a major push to genotype our materials as well as phenotype them. That is there is a big push for that if this plan comes to fruition. So in conclusion, the NPGS is one of the largest gene banks in the world, and the world uses the material that we have, which is great. That's our purpose, is to support not only US agriculture, but agriculture around the world. The NPGS has challenges that every single gene bank around the world faces, insufficient resources to do the job right. Happily, though, we have developed this plan, and Congress will be reviewing it. And so we're all crossing our fingers that the future is going to be bright with opportunities to strengthen our operations and the science behind what needs to be done to do good gene banking. All right, and with that, I'm not sure if I have time for questions, but happy to take them.
We can take a couple of questions.
Hi, Stephanie. Great talk. Great to hear that you guys are getting some funding or going to Congress to request more funding. I think that's really important, and the mission that you guys have is really important. With the distribution across all the different sites, what's the best way to find out where a specific crop is? Do you know what I mean? Like if you've got certain centers of excellence, what is it? Can you get that information through Greene?
Yes, you want to go to the section that basically there's a map that shows where all of the germplasm repositories are across the United States, and then it'll give a breakdown of what is the crop that is held at the at that site. Mm-hmm. I think to effectively use the material in the NPGS, it's critical to reach out to the curator of that collection.
Okay.
So it's good to go to the actual, there's a place on Greene where it says repository websites, to go to those websites to find out who curates what. You can also, and it's kind of clunky, but if you have an account for Greene, there is a place that says reports. You do have to be a registered user, but it's free and it takes 2 seconds to sign up. And, mm-hmm. and you can actually do a query. If you go into the reports section, there's a specific query that says what curator, to find the curator for the specific genus and species that you're interested in. And so you can go ahead and use that little clunky query to find out who curates what on a species level.
Okay, great. And I probably know the answer to this given the funding, but like with all the different growing systems that are coming online, say for instance, vertical farms, is there an effort to like kind of look at how these plants perform in different environments, for instance? Or is it kind of just considering G by E, for instance, right? Like are you sending material other places to see if it performs well, kind of similar to Roland, or in different growing environments, like a vertical farm?
It's, yes, we are doing that. And again, it's individual curators that are forming, they partner with universities, mainly universities, other ARS people, to send out to do multi-location evaluations. And it's really, it depends on the curator and what the resources are to evaluate germplasm at this point in time. Each gene bank has its own budget. Some gene banks have less money than other gene banks and less resources. Some curators have been particularly effective at getting extramural funding to support these kinds of evaluations. Again, partnering with others to conduct these kinds of evaluations. So again, the easiest way to find out what's going on is to focus on what is the crop you're interested in, and then reaching out to the curator with your questions, and they can pretty quickly fill you in as to what they're doing, what type of evaluation data is out there.
Okay, great. Thank you.
So let's thank our speaker again. Just a reminder, next we have Dr. Dior Kelly and Dr. Alex Bucksh presenting next week. Thank you all for coming, attending, and presenting.